Our next and only speaker for this session is Professor Lindsay Hall, who is a group leader in the Early Life Microbiota Host Interactions Lab at the Quadram Institute. So Professor Hall and her team focus their research on defining microbe and microbiota interactions during early life development.
So afternoon, everybody. Fantastic session this morning. And really appreciate all the speakers who have actually already introduced kind of some of the concepts that I'm going to talk about. And as Sophie mentioned, me and the team, we look at the gut microbiome, but we specifically look at the early life developmental window. And what I want to do kind of the next 30 minutes or so, and I'll just make sure that I am within that time frame, is give you a little bit of background into kind of these microbes and when they colonize infants. And specifically some of the work where we've been introducing pro and actually prebiotics to at-risk infant populations, including premature babies. And I'll give you a little bit of background about that, as well as some of the data that we've done for that clinical study that will hopefully be of interest. And of course, very happy to answer any questions. After the presentation, I'll also pop on to discourse as well to answer any questions that anybody might have. So without further ado, so we're particularly interested in understanding the gut microbial kind of ecosystem at the first, I guess, hours, days and weeks post-birth, because this is when you start to collect your hopefully beneficial bacteria. And actually, when we think about early life and these microbial ecosystems establishing, we really think within kind of 1,000-day window. And this is a critical time period, as we well know, for kind of just host physiological development. So, priming the immune system, setting up the metabolic system, and of course, a whole host of other kind of physiological parameters, including brain function. But this is also when we acquire our first microbes. So it really represents the time period in which we can either kind of beneficially change the microbiome, or we can also potentially perturb these initial microbial communities that may mean that there's both short and longer term health consequences for the baby and as they grow older as well. So this is very much kind of this symbiosis, this partnership between host and microbe, and it starts basically post-birth. So I won't go into too much detail here because I'm conscious of kind of the time. But there's a number of different factors that can potentially interrupt or disrupt the transfer of microbes from mum to baby, and also the establishment of these microbes in the infant gastrointestinal tract, basically shortly after birth. So depending how a baby is born, so if a baby is born by cesarean section or vaginal delivery, basically this can interrupt the transfer of vertical transmission of microbes from mum to baby. And actually, characteristic of that would be the fact that C-section babies also sometimes have an absence of beneficial bacteria, including bifidobacterium and Bacteroides as well. Preterm birth, I won't talk about that because we're going to come back to that. And then the other one that's classic, which we've already heard, but specifically for the early life window, is diet. And what do I mean by that? Well, we're talking about milk. So we're talking about breast milk, we're talking about formula milk, or we're talking about a mixture of both. And I'm going to talk about this more specifically as I go through the presentation as well. And then the other thing that is really important in early life is that many babies get antibiotics. And antibiotics do not discriminate between beneficial bacteria and the pathogenic bacteria that they're hopefully trying to kill. And this has a massive impact on ecosystem structuring. And again, this is something I'm going to touch upon as we move through the presentation. So we're just going to dive in to think about preterm birth. And why should we care about this? Why should we care about preterm babies? So preterm babies are at risk. And actually, they represent 1 in 9 live births globally. And in fact, that number is rising. And in some countries, that number is actually even more common than what we have in the UK. So preterm babies are born or defined as born less than 37 weeks gestation. And they're often very low birth weight. So kind of about 1.5 kilograms. Some of our babies on the studies that we've been doing are much smaller than that, down to even 500 grams. But to put that into context, a full-term baby is about 3.5 or 4 kilograms. So these are small babies. Because these babies, they're obviously born prematurely, they're born early. The gut and lots of other organ systems haven't had the same time to develop because they've not been basically in utero for long enough. And they also have an underdeveloped immune system in comparison to their full-term baby counterparts. Unfortunately, these preterm babies are exposed to some of the microbiota modulating factors I kind of just introduced on the first slide. So many of them born by cesarean section, so interrupting transfer of beneficial microbes from mum to baby. They reside in sterile hospital environments, so neonatal intensive care units, and we'll talk a little bit about that in a moment, about what that might mean for the early life microbiota. And unfortunately, these babies are often prescribed probiotics, not probiotics, I'm going to come on to that, antibiotics, so the other flip side of the coin, where actually up to 75 to even 100% of preterm babies are given antibiotics. And actually in the UK and in many other countries, if a baby is born less than 1.5 kilograms, or often low birth weight or low gestational kind of age, then they're automatically prophylactically given a course of benzoyl penicillin and gentamicin at birth. So if they were born by baby, natural birth or vaginal delivery, these antibiotics might be negatively impacting these new microbial communities. So obviously the hospital environment, neonatal intensive care units, the hospitals, the doctors, the clinicians, the nurses, the healthcare practitioners that are looking after these babies do obviously an absolutely phenomenal job. But in these units and these preterm babies, they don't have a normal gut microbiota. So if we take a poo sample from a preterm baby and we compare that to a baby that's full term and not in a neonatal intensive care unit, what do we see? Well, we often see these preterm babies are missing some key beneficial bacteria, including bifidobacterium, which is going to be the focus of the talk, and it's one of our favourite groups of bacteria. And they're often colonized by potentially pathogenic microbes, including kind of Klebsiella, E. coli, and enterococcus. And importantly, and we'll talk about this briefly, is that these pathogens are often antibiotic resistant, which is potentially a disaster. So the preterm gut is radically different than the full-term gut. And this is really important because one of the main functions the microbiota plays for host and host protection is something called colonization or infection resistance. So the microbes that are in the gastrointestinal tract, they actually compete for nutrients, they compete for niches, so that might be in the gut epithelial barrier here, and they also produce kind of novel antimicrobials that actually can basically inhibit or kill potentially pathogenic microbes. So this is kind of colonization resistance is what we want, but this is nearly always much reduced or even absent in preterm babies, and that's completely due to the fact that they are missing some of these key beneficial bacteria. And this is really important because this lack of colonization resistance in the preterm gut is linked to an absolutely devastating disease called necrotizing enterocolitis. And this kind of aberrant or this disturbed microbial colonization in preterm babies means that they have a heightened risk of NEC. And NEC affects between 5 to 15% of preterm babies. It's got a really significant mortality rate of up to 40%, and that can be much higher in different neonatal intensive care units and depending on where you are in the world. And if these babies do get NEC, but they survive, there's really serious long-term health consequences. And this is because, just to give you some of the clinical symptoms here, these babies present with basically necrosis in their bowel. And the neonatal surgeons will have to go in and remove the necrotic part of the bowel and basically stitch together the healthy parts. But you've just taken apart a preterm baby's gastrointestinal tract, which means that there's less kind of surface area for absorbing nutrients, which has longer term health implications for kind of brain development and just overall physiological development. And these babies will go downhill very rapidly within 6 to 8 hours. So they will present relatively normally for a preterm baby, but then they will start to present with this descended abdomen, which if you look under X-ray is something associated with pneumocystis intestinalis, which is gas cysts in the bowel wall. And this is all because we have lack of colonization resistance and overgrowth of potentially pathogenic microbes in the preterm gut. So what I want to kind of walk you through kind of this afternoon is, so preterm babies don't have the right kind of gut microbes, but can we actually boost or restore their gut microbiota into something that looks more like a full-term baby and hopefully reduce the incidence of necrotizing enterocolitis. So what we have here is this is what we want. This is what we want, a healthy full-term infant gut with resilient colonisation resistance. So we want the right microbes that are competing for dietary components coming into the gut. We want them competing for niches within the gastrointestinal tract. And we also want them to produce novel antimicrobials that will inhibit the overgrowth of these pathogens linked to necrotizing enterocolitis. And one of the main kind of groups of bacteria that we're really interested in to kind of be the right bacteria at the right time is bifidobacterium. And this is kind of where our microbial pioneer or our microbial keystone species comes in. And of course, I think most of you will know or understand that bifidobacterium, or many different species, are known to be probiotic. And actually, they're known to be generally recognized as safe, which means that we can kind of move into the clinic very quickly. So just to give a little bit more background about bifidobacterium. So in a full-term baby that's kind of vaginally delivered, these are some of the first microbes that are transmitted from mum to baby. And they're really important for ecosystem structuring in the first kind of, time phases after birth and longer term as well. Importantly, they kind of overlap with when the infant immune system is programming. And we've got other work where we've actually showed that bifidobacterium programs specific aspects of the immune system, but I won't go into detail here. These bacteria are exquisitely adapted to the diet, and specifically within early life, the milk-based diet. So breastfed babies are known to be dominated by bifidobacterium, whereas formula-fed babies might have bifidobacterium, but the overall levels are much lower. The question about why that relationship happens is something that I'll kind of cover. We'll come back to that. Already kind of mentioned this, but a number of different species can be used as probiotics, and they're known as GRAS, or generally recognized as safe. And this is really important because a number of diseases are associated with reductions in bifidobacterium abundance in the gastrointestinal tract. And unfortunately, bifidobacterium is one of these kind of groups of bacteria that are very susceptible to some of the factors that I've talked about. So antibiotics, diet, and also how a baby is born. And importantly, for the kind of the context of this presentation, bifidobacterium levels, I've already alluded to this, are much, much, much lower in preterm babies. And this is associated with a higher risk of developing necrotizing enterocolitis. So this is kind of where I'm going to dive into the clinical study that we've done on this area to give you a little bit of background about why choosing the right bacterium with the right diet is really important in at-risk infant populations, including preterm babies. So this is our Bambi study, so baby-associated microbiota of the intestine. I'm not going to kind of talk through all the data, don't worry, but this is an observational clinical study that was done in 4 different neonatal intensive care units in England. And it comprised 2 groups of preterm babies that were kind of under having routine clinical care. So we had a group of babies that were probiotic supplemented, but I'm going to refer to these as BIF-lacto groups or BIF-lacto supplemented babies. So these are babies that got given this particular probiotic here, which includes Bifidobacterium bifidum and Lactobacillus acidophilus. And then we have our control babies that did not receive probiotics. And this particular study was led by 2 fantastic people in the team, Christina when she was a PhD student and Matthew as the postdoc. So Christina's moved on from the lab, but Matthew's still in the lab and doing fantastic microbiome clinical studies. And what we did here was we collected poo samples or stool samples from the preterm babies when they were residing in the neonatal intensive care units, kind of right up to 100 days at 4 different time points. And the data I'm going to show you as we move through is this kind of data cohort. So, and this has already been introduced, but obviously what we're dealing with here is probiotics. So live microorganisms that when administered in adequate amounts confer a health benefit on the host. And hopefully I'll convince you in the context of preterm babies and probiotics in NEC, probiotics work. So what we did to kind of profile the gut microbiota of our preterm babies was we used 16S ribosomal RNA amplicon sequencing. And I'm not going to go into the massive detail here. I think the important thing is basically for you to start looking at the microbiota signatures that we're seeing in our two groups of preterm babies. So this is just sample one. So this is the babies between 0 and 9 days of age, so very quickly post-birth. And what we're showing here is an NMDS plot. And this is something we use a lot when we do microbiome profiling. It kind of really helps, I guess, with showing the overall complexity of the gut microbiota, but in a way that's relatively easy to digest. So the take-home messages from this particular figures is our blue triangles are our supplemented babies, and our red squares are our control babies. And each of these individual points are individual babies. So you can see this is a very good number of babies we've done a study on. And this is time point 1. And you can hopefully already see from this plot that there's really quite a significant divide between our two babies. So our supplemented babies seem to be kind of moving away from our control babies because they have bifidobacterium, whereas our control babies don't have bifidobacterium, but they have potentially pathogenic bacteria, including Klebsiella and Staphylococcus. But let's look at this in a little bit more detail. And if we look at this kind of on a bar graph here, we can see this kind of more clearly. And again, this is time point 1. So we see that our supplemented babies versus our control babies who have not had the probiotic, they have lower levels of these potentially pathogenic microbes. I've mentioned some of these, but also Enterobacter and Escherichia, so E. coli are lower. And importantly, the two kind of groups of bacteria that were in the probiotics, so Bifidobacterium and Lactobacillus, are higher. And this is only time point 1. We have published this, and I think there was a link on the other slide, so please go and have a look at the data if you so wish. But we saw a very similar profile right across the study period here. Now, let's look at Bifidobacterium in a little bit more detail. One, because we like Bifidobacterium, but I think there's some really important take-home messages here about why some of the factors that affect the gut microbiota might be affecting the Bifidobacterium, but more specifically, how we might be able to boost colonization of the probiotic for preterm baby health. So if we look right across kind of the sampling time period here, and we look at Bifidobacterium, and we're comparing our control versus our supplemented babies, we can see at the early time points, our supplemented babies have much higher abundances or levels of Bifidobacterium in their stool samples in comparison to only relatively few control babies that have some Bifidobacterium. So the control babies do naturally become colonized with Bifidobacterium over time, but this happens much later. And this is really important because the time window for necrotizing enterocolitis, the average time of when this disease happens is within the first 2 weeks. So even though we get this natural colonization, it's potentially too late to stop NEC, which is why we need to actively supplement these babies. And then if we go and think about some of the microbiota modulating factors that I talked about previously that might be associated with kind of differences or disturbances in the preterm gut, and we do this kind of with various statistical kind of models that we apply to our microbiome data, is that if a baby's being supplemented, yes or no, is the most significant variable explaining the variance, explaining the differences between our babies, which shows that the probiotic supplementation is doing something. Surprisingly, it looks like delivery method actually doesn't seem to be contributing that much of the variance. I'm going to talk about this on the next slide. But birth weight has a massive impact on kind of overall microbiota profiles. And this is really important in the context of the most at-risk babies of NEC. And I'm going to talk about this in the next slide. So what are the impact of these variables on Bifidobacterium abundance in the preterm gut? So I told you that C-section babies and vaginal delivered babies have quite different gut microbiotas. But actually, when we look at Bifidobacterium abundance and we kind of divide this by delivery mode, surprisingly, we don't see that much of a difference. But let's look at the next graph here. So if you remember, I mentioned that many preterm babies prophylactically given antibiotics. So actually, for many, many babies, even if they were born by natural delivery, they get given antibiotics almost immediately. So if they did have BIF transfer from mum during natural childbirth, the slate has been wiped clean. And this is basically why we don't see a difference in birth mode. But importantly, bifidobacterium is not antibiotic resistant. And this means that antibiotics really kind of have an oversized negative effect on bifidobacterium levels in the preterm gut. But what about some of these other pathogenic microbes associated with necrotizing enterocolitis? Well, unfortunately, we don't see the same problem. So in this case, what we see is if a baby's been given a course of antibiotics, we don't see a reduction in these potentially pathogenic microbes. And other work we've done is actually shown categorically that these are often multi-drug resistant, which is probably why they don't care that antibiotics are being given. Another important finding is the fact that if a baby's born very early, and if a baby's born very low birth weight, they actually have less bifidobacterium colonization in their gut in comparison to other preterm babies that might be born a little bit later or are indeed higher birth weight. And that's really, really important because those babies most at risk of NEC are extremely low birth weight babies. And this is the group of babies we want to get bifidobacterium in because they're the most at risk of NEC. And in fact, this is something we're following up in other clinical studies at the moment. So one of the things that I think maybe some of you have kind of read or you might have heard kind of the news or even in scientific articles is that a lot of people say that probiotic supplementation, if you stop taking it, it basically just gets pooed out the other end and you don't get longer term colonization. But this is actually not what we see in the preterm gut. So the babies, although they were supplemented daily with a probiotic in this clinical study, we actually stopped the probiotic supplementation after they'd reached kind of the equivalent of 34 weeks gestational age. So they got to a point where the probiotics were stopped. However, what was still really interesting was when we looked at the baby's poo after the probiotic supplementation was stopped, we still saw really high levels of bifidobacterium. So why is bifidobacterium sticking around even when the probiotics stop being given? This would suggest active colonization, but how do we categorically show that this is the case? So if we actually look kind of delving a little bit deeper rather than just looking at bifidobacterium, but if you remember, the species of bifidobacterium in the probiotic was Bifidobacterium bifidum. And we can see that this is almost the only species that we find in our supplemented babies. We hardly see any of our control babies with this species. So this would suggest that, yes, it's the probiotic species that we're seeing and it hangs around even after the probiotic is stopped. But we're microbiologists in the team, so we always want to look at this in a little bit more detail. And the gold standard way of doing this is basically going back to baby poo, isolating whole bacteria out of that poo, and then actually sequencing their complete genome, and then comparing basically the DNA sequence of what we get out of the baby poo with the reference strain. And in this case, of course, that's the probiotic that we've given. And actually, if we do a whole load of analysis, what we can see here is it's the same DNA sequence. So we can say, I guess, conclusively, that even after the probiotic stopped, we can still detect the exact same probiotic strain in preterm babies after. So the question is about why do we see this? So basically, why do we see this longer term colonization? What is it about what we've kind of done with the preterm babies that really facilitates this long-term colonization? And actually, this is the miraculous biological properties of breast milk, and specifically the sugars that are found in breast milk called human milk oligosaccharides, which, as we've already heard, actually represent an important group of prebiotics, because these complex sugars are only digested by certain microbes, and in fact, these human milk oligosaccharides are the favorite food of bifidobacterium. And what we've done here in this case, I'm going to hopefully convince you just in a couple of slides time, is that what we've paired a microbiota therapy, so a probiotic, with a diet to facilitate this longer term colonization. And this is because human milk oligosaccharides are not actually broken down or digested by the baby. These complex sugars pass undigested into the colon, where actually the resident bifidobacterium will do that digestion for the baby. And then they produce specific metabolites, specific compounds that then in turn feed the baby. And I'll talk about some of these in a moment. But some of these include short chain fatty acids, which are these beneficial microbial derived metabolites. So just to kind of convince you, why are bifidobacterium and breast milk, why is that kind of the perfect pairing? And specifically for this particular study, the species of Bifidobacterium bifidum has all of the kind of enzymatic machinery that's capable of digesting these complex sugars. And actually, we can see this if we just do a growth curve. So if we take our Bifidobacterium bifidum and we grow it in breast milk, we see it loves breast milk because it grows to really high levels. And we can also see this is whole breast milk, but we can also see that in response to specific human milk oligosaccharides, specific sugars, we also get this boost in growth. And this growth is related to the fact that we've got the genes in the Bifidobacterium genome that enable the Bifidobacterium to digest these complex sugars. And just something I think that's really important to highlight here, because some of you, I might be already maybe thinking about some of the questions that you might be asking. What we did in this particular study, which is why we think we saw such excellent colonization in our supplemented babies, is that nearly all of our supplemented group got also donor breast milk or express breast milk from mum. Now, donor breast milk is pasteurized, but actually even the pasteurization process, it enables these human milk oligosaccharides, these sugars, to remain intact. And in fact, potentially you might think it's a confounder if the baby's had express breast milk or donor breast milk, but actually under these circumstances, what we see is we see all of the babies basically get donor breast milk to boost express breast milk from mum. And actually, if we look at the babies and we go back to these NMDS plots we showed, we can't separate the babies. So it doesn't matter if the babies had donor breast milk or express breast milk, we see very similar levels of colonization of Bifidobacterium, which is really important because it highlights the importance of donor breast milk and the fact that we need more donor breast milk banks to be providing this kind of gold standard nutrition to these at-risk babies. So what we've got is we've got this boost in growth of Bifidobacterium, which is because they're digesting these complex sugars. Just last couple of slides is what does this mean kind of at a functional level? I mentioned about the fact that this HMO kind of digestion means that Bifidobacterium produce active metabolites that are beneficial for the baby, but what are these metabolites? So this is where we kind of use metabolomics to kind of complement some of the microbiota profiling methods using kind of DNA sequencing. And what we have here is we have kind of just a spectra, an NMR spectra that shows some of the metabolites that we get in our babies. So this kind of top half of the plot is our supplemented babies and the bottom half is our control babies. And all of these peaks relate to specific compounds or metabolites that we can detect in the preterm baby poo. So don't worry too much about this, but the take-home message is supplemented babies have higher levels of short-chain fatty acids, including acetate and lactate. And importantly, these short-chain fatty acids are acidic. And actually, Christina pH'd preterm baby poo and actually found that the control babies had about a neutral pH of their stool sample, but this was reduced to about pH 5.5 in our supplemented babies. And this is important because a lower pH actually inhibits growth of pathogenic bacteria, including Klebsiella and E. coli. So actually what we're seeing here is that the supplementation with probiotic is completely changing the environment of the preterm gut. And this actually we already saw potentially is why we're seeing these reductions in pathogenic bacteria associated with NEC. And if we correlate microbiome profiles with these metabolites, and this is just basically kind of showing you what I've already explained, Bifidobacterium is associated with higher levels of short-chain fatty acids, acetate and lactate, but lower levels of human milk oligosaccharides. And why are we seeing that? The BIF is actively metabolizing these sugars. The fermentation end products are these short-chain fatty acids, which are acidic, which reduce the pH. So this is kind of what we see from a microbiome profiling perspective. And of course, when we speak to our kind of, healthcare colleagues, our clinical collaborators, they don't really care that much that we're changing these microbial communities, although as a microbiologist, it's really important to show this data. But actually, this was a fantastic kind of clinical collaboration with Paul, consultant neonatologist, and Claire, who was a trainee clinician in the lab. And what we were able to show is that after we started giving routine probiotics to our preterm babies, we were able to reduce the incidence of NEC by 50%. And in fact, we were able to also reduce late onset sepsis and all cases of mortality in our preterm babies. And that's really, really important. So that's like fantastic to see kind of a clinical outcome by probiotic and right diet. And in fact, based on this data that we've kind of produced together, we've convinced other neonatal intensive care units in the UK to start doing routine probiotic supplementation, and other neonatal intensive care units actually globally as well. So it's really exciting to see this impact. But we've not had 100% reduction in NEC. So we've also been doing some work, and I won't talk about this, but we've also been trying to kind of come up with new novel ways of rapidly diagnosing necrotizing enterocolitis and these more at-risk babies. And this is using kind of some new sequencing technologies where we're able to take a poo sample, extract the DNA, profile the gut microbiome, and we're able to tell the clinicians what antibiotics that the bacteria that are causing NEC might be resistant to, so they can choose the right antibiotic. And again, if you want to read that, then please go ahead. That's just the paper there. So I won't labor the point too much because I think we've already covered this. The preterm gut microbiota is normally kind of compromising some potentially pathogenic microbes, but we don't want this. So one way to kind of beneficially change the preterm gut is to give probiotics. And specifically here, we were able to show giving a specific probiotic, we were able to beneficially change the gut microbiota into one that was bifidobacterium rich, which is what we want to see. However, not all of the babies had really high levels of bifidobacterium abundance, and this was linked to antibiotic usage and if the babies were born kind of very early. And this is a group of babies we're kind of now focusing on really quite carefully because they're most at risk of NEC. But we do see this really lovely longer term colonization in our babies. And that's because we have this active metabolism of breast milk sugars by bifidobacterium, which produces these short-chain fatty acids and reduces the pH. And importantly, all of this together means that we actually reduce necrotizing enterocolitis in our preterm babies. But I think what's really important to highlight here, and I think it's already been highlighted by fantastic talks earlier today, for this we need to think about microbiota strategies that are coupled with the right diet to kind of promote this efficient colonization longer term so we get really beneficial health impacts. And last but not least is our is obviously a fantastic team that I work with, because all I do is respond to emails and try and get some money for these fantastic people to do the work by our fantastic funders. And just one final slide, I'm just conscious of time, is what's next. So we've been kind of looking at preterm babies, but we realised we maybe need to go a wee bit earlier. So actually a longitudinal cohort study that I'm chief investigator of that's nearly finished with recruitment and has been understandably somewhat impacted due to the ongoing pandemic COVID, is we've actually been looking at pregnancy in early life. So we've been sampling mum and baby right through kind of trimester 1, 2 and 3, birth and post-birth. We've got all of the dietary information as well. And these are healthy mums and babies. And what we're wanting to do here is understand healthy diet and microbiome signatures so we can start to develop actually new maybe probiotics or diet strategies for mums that are maybe more at risk of preterm birth or other pregnancy complications, and to beneficially promote babies' health longer term as well. And that's everything from me. And thank you very much for listening, and very happy to answer any questions that you have. Thanks.
Thank you, Lindsay. I think this work is just absolutely fantastic. And we do have a few questions for you. Can I start by just saying, so you supplemented, the probiotic you used had bifidobacterium and lactobacillus. And you've very clearly demonstrated that the bifidobacterium colonized the preterm gut really well. But what about the lactobacillus? Did it colonize as well? And if not, why not?
Yeah, so that's, yeah, so some of you eagle-eyed people watching the presentation may have kind of seen that the lactobacillus levels were quite low. And in fact, you can go to the paper to have a look at that a bit more detail. So importantly, lactobacillus is kind of in different species of lactobacillus are often kind of incorporated into probiotics, but actually people think that lactobacillus is found really high levels in the preterm gut, or rather just in the infant gut, but actually we don't normally see that. So lactobacillus is fundamentally important in the vaginal microbiome, but maybe not so much important in the gut. However, it could be that lactobacillus actually comes in and is found at higher levels very early on, and then it's lost, but without that, we wouldn't get that really good colonization of bifidobacterium. Or another point is that we're obviously looking at poo samples, because of course doing any other fecal sampling from these preterm babies or doing endoscopy is completely and utterly a no-no. So lactobacillus actually quite likes to live in the small intestine. So it might just be that we're missing the lactobacillus signature due to the samples that we've been actually taking from our preterm babies.
Oh, I see. I noticed in one of your slides that you talked about the reduction in pH and how that deterred pathogenic bacteria. Is there any other mode of action that you think the bifidobacterium might be doing to reduce NEC?
Yeah, so we were obviously, again, we were fairly limited with what we could do. I mean, we'd actually hoped to maybe take blood samples from the preterm babies, but of course this is very difficult to do, and also making sure we kind of get mum and dad on board in terms of wanting to actually, consent. They're fine to give away poo samples. Anything more invasive, it's maybe not great. So one thing that we have done with other studies, and that's kind of mouse models as well as other clinical studies where blood is kind of more readily available, is we actually know bifidobacterium is fundamentally important for programming the immune response. So in this case, what we think we're also seeing is we think bifidobacterium is kind of boosting anti-infection responses by the immune system. So we've kind of got a double whammy. There's that direct inhibition due to pH coming down, but we've also got kind of boosting of immune responses, which will also potentially fight off these neck-associated pathogens as well.
Fantastic. So how important do you think is pairing the right diet with the right probiotics in preterm infants?
So it's really, really important, because there's been a couple of studies done that have done bifidobacterium supplementation, and they've not seen a concurrent reduction in the rates of necrotizing enterocolitis. And the question is, why? And that's basically because even though I talked about bifidobacterium and its favorite food being breast milk, and specifically these human milk oligosaccharides, not all species and strains of bifidobacterium encode the right kind of enzymes to break down these sugars. So if you don't choose a bifidobacterium that can break down these sugars, they won't hang around, and therefore you're probably not going to get the beneficial effects. So actually, it's really important to understand kind of the underlying, I guess, DNA of your probiotic and what it can and can't do, and then think about pairing it with the right diet. So for example, we've heard more kind of on the adult side of things. There's some bifidobacterium that have got machinery able to digest resistant starch, but not all of them can do it. So if you're going to do that kind of combo, you need to pick the right bifidobacterium with the right type of starch. So it's kind of always thinking on both sides of the coin, and that's really, really important consideration.
Wow, that ties in really nicely to a couple of questions that we've had. Firstly, from Molly, and she says there are some new products containing a mixture of bifidobacterium strains as probiotics for babies. She wants to know, do you think there would be benefits to introducing multiple strains versus just one in probiotic therapies?
Yeah, great question. So, and in fact, this is kind of something that we're kind of moving more towards as well. So if you kind of profile a healthy, full-term, vaginally delivered breastfed baby, it's very rare you will find one type of bifidobacterium. You normally get these mini bifidobacterium ecosystems. So actually, probably it's more beneficial to maybe give a couple of different bifidobacterium. However, again, you just need to make sure that that combination works well together, because sometimes you get bifidobacterium that might actually inhibit the growth of another bifidobacterium. So you need to kind of do all those studies to make sure you've got the right combo before moving forward. But yeah, I think probably more of that cocktail is probably more beneficial than a single strain.
For the majority of cases, then.
Yeah. Yeah.
So that again, thinking about breast milk, what's the difference? Oh, this is from Amy. What's the difference in the preterm baby that doesn't receive breast milk after supplementation? Or do all very preterm babies receive breast milk as standard policy?
So for our study, kind of the normal kind of healthcare for the preterm babies is hopefully breast milk from mum. Although if baby's born very premature, the mammary gland might not have developed. It might take a little bit of time for that milk to kind of come through. So this is where donor breast milk banks are really, really important. So nearly all of our babies got breast milk, either express breast milk or donor breast milk. However, not all neonatal intensive care units have access to that donor breast milk. So if you actually look for other studies where they've done probiotic supplementation and it's been formula given to the preterm babies, that's probably one of the other reasons why you don't see kind of that normal natural colonization of bifidobacterium. You're not giving it something it likes to eat. And then also that's probably why some of those particular studies, we haven't seen that reduction in NEC either or other health outcomes. So it's really important. I think breast milk, which I probably didn't say, doesn't contain one type of human milk oligosaccharide because that would be too simple. It contains multiple human milk oligosaccharides. So even if you have a formula with an individual human milk oligosaccharide or even two, it's nowhere near as kind of rich as that gold standard nutrition that's breast milk. So that's why these breast milk banks are really, really important. And hopefully we're beginning to see more of these in the UK and also kind of across other kind of European countries and globally as well. But we need far more of a push for this.
Right. I've got a lovely question here from Claire, who's asking, do you also see a significant link between babies with low bifidobacterium and allergies coming up in later life? I don't know if that's something you can comment on, Lindsay?
Yeah, a great question. So other people have done this study where, yes, you're completely right. So bifidobacterium, because you find it really early and it's really important for priming the immune system, if you don't have it at the right time, low bifidobacterium is linked to higher rate of allergies. That might be atopic dermatitis, so eczema, but also kind of asthma as well. And that's probably because you're missing BIF programming the right aspects of the immune system. So one thing we definitely like to do with our preterm babies that have got our high bifidobacterium and then our control babies that don't, is we could then do a follow-up study to look for incidence of allergy and to link that back to give more evidence to say, let's give BIF back, let's program the immune system, and then we've got the benefit of reducing these horrible diseases like NEC, but we've got these wonderful longer-term health benefits as well.
Brilliant. I just have one more question. So you've mentioned that various hospitals have taken up this now as a sort of standard policy based on the evidence that you and your collaborators have produced. What I'd like to know is, what's stopping that from being rolled out nationally and internationally? Is it that there's more evidence required, or is it a lobbying issue?
It's complex, Becky. So there was definitely part of the fact that there was a number of kind of neonatologists that didn't feel that the scientific evidence was robust enough. So there wasn't robust enough kind of clinical trials in the area. I think there's a huge body of evidence now that indicates that the probiotics, are good. But I think one thing we're missing at the moment is everybody's using different probiotics. Some of them work, some of them don't work. I can probably tell you why as a microbiologist, but that's why we need people to be more kind of in tune with thinking about the microbiology side of things. So I think there's definitely a push, but I do know that there's been some other bigger studies that haven't shown a benefit, so that kind of makes people a bit worried. We didn't see any negative health consequences at all with our routine probiotic supplementation, because these are at-risk babies and giving a live bacteria might be considered a risk. We don't see that, but that's something as well that I think is maybe stopping other people working in NICUs from maybe giving it out routinely, although considering how many babies' lives we're saving. Yeah. But this is fine. We just need to bring everybody along with us. And that's why we need to be kind of doing these studies. We need to be doing this further. And in fact, this is why we're now doing Pearl. We're able to give it mum, and mum doesn't even have preterm birth, which is fab because the baby's not in NICU. But this is why we're actively working in this area and doing the basic science to actually inform kind of clinical work and science policy as well.
Yeah, and in return, then understanding the evidence that's out there and the differences between them. That's great. So will you be sharing your slides with the live viewers today, Lindsay?
Yeah, that's fine. I'll just tweak them a wee bit so I can take out everyone's photos because they won't be necessarily happy with sharing them. But yeah, and then there's links to the papers and other stuff. So yeah, happy to share that bit.
Perfect, because I'm sure some of our viewers would like to follow up on some of the brilliant resources that you shared with us. So I'd just like to say thank you ever so much for your time, Lindsay.